The 69th annual meeting of the Gulf and Caribbean Fisheries Institute was held in Grand Cayman in November 2016 and was hosted by the Department of Environment of the Cayman Islands. The opening address was presented by Dr. Brice Semmens, who's an assistant professor at Scripps Institution of Oceanography at the University of California in San Diego. Dr. Semmens has been involved with GCFI for the last 20 years, having attended his first GCFI meeting as a PhD student in 1997 and serving on the board of GCFI since 2015. Dr. Semmens' research focuses on fisheries conservation science and quantitative methods for resource assessment. For the last decade, he's led the Grouper Moon Project, a research partnership between the Cayman Islands Department of Environment, Reef Environmental Education Foundation, and Scripps Institution of Oceanography. It's one of the world's most comprehensive and long-term multi-species fish spawning aggregation monitoring programs.
I'll start with the supposition that aggregating fisheries in the Caribbean have been relatively poorly managed. And there's no better example of that than the Nassau grouper. It's the classic, iconic example of an aggregating species that's really tanked, honestly, over the last century. If you look at catch records from 100 years ago, this species was the most important finfish fishery, certainly one of the top finfish fisheries throughout the Caribbean basin. And now it's essentially a footnote. There's very few places left with any appreciable number of the species, and even fewer still that support an active fishery. And we know why this is. We know why aggregating species are difficult to manage, and in particular why Nassau grouper are doing poorly. And it turns out that the reason goes back to basic fishery science. When we seek to manage a fishery, we often have to rely on fisheries data. We rely on metrics of abundance that are derived from catch. So we assume that the amount of fish that are available for us to catch, the amount of fish in the water, is proportionately and directly related to our catch per unit effort. So if I catch a lot of fish per hour fishing, there's a lot of fish in the water. If I catch less fish per hour fishing, there's less fish in the water. And if we make the simplifying assumption that those things are monotonically correlated or related, then we have, through the fishing data, we have an active record of what's going on in the fishery, and we can be proactive about how to change fisheries management in order to conserve the species. But this simplifying assumption doesn't always hold. And there's two ways where it doesn't hold. One is if we have something called hyperdepletion. And in hyperdepletion, you have a rapid decline in catch per unit effort with only a shallow decline in abundance. This isn't a problem for conservation of a species. It's certainly a problem in terms of managing the fishery appropriately and getting the most out of it. But what's really a problem for conservation of the species, for making sure that the fishery sticks around into the future, is hyperstability. And this is what aggregating species suffer. In hyperstability, you see stability in catch per unit effort. Your number of fish that you're pulling out of the water per unit time remains relatively constant despite deep declines in the population itself. And it's not until you get to the very bottom end of that population, when things are really bad, that you start to see declines in catch per unit effort. But at that point, honestly, it's too late. It's too late. And so you are left with, okay, how do we tweak fishing to maximize? You can't do that anymore. Now you need to start thinking about rebuilding plans. We need to stop fishing. We need to recover this population. And so this disconnect between abundance in the water and catch per unit effort causes our aggregation fisheries to do very poorly. So that's happened here in the Cayman Islands. It's happened everywhere in the Caribbean, honestly. But if we look at the history of fishing grouper holes in the Cayman Islands, they've been fished for over 100 years. Historically, they were fished with rowboats and hand line right the way up into the 70s. And it wasn't really until the 70s that we moved here in the Cayman Islands from a subsistence fishery, getting fish onto the table locally when in season, to more of a commercial process, where you started getting rigs coming from Jamaica and buying fish off of local fishermen. And that process really started in the early 70s, where you were getting $1 a pound for fish. A good catch during those days with hook and line would be roughly 300 pounds a day, or let's call that 30 fish or so. But once the Cayman Islands started commercializing that process, now there was incentive to take as many fish as possible because you could sell those. And that, at the same time, there was advancements in the technology. Things like everybody started using outboard engines, and those things all culminated in a rapid increase in the amount of take from those aggregations. And by the 1980s, early 1980s, we'd gotten to the point where the Cayman Islands fishermen were starting to notice, look, our catches are declining. And they went to the Cayman Islands Department of Environment and said, hey, help us with this. This is not good. Things are not as good as they used to be. And at the same time, during that window of the 70s, the Cayman Islands economy started to shift from one of mainly resource use to one that was driven by tourism, and today remains largely driven by tourism. And that shift was around the same time. And how many of you have cats? Who has cats? Okay. So I would argue that Nassau grouper are important for the tourism economy because they're like cats. So if you open a cabinet, or if you're messing with something on the bottom, a Nassau grouper is gonna be quite keen to figure out what you're doing. So here's Dr. Steve Giddings helping us set up some equipment out on the grouper hole, and this grouper is certain that there's something delicious there. And as a diver, you can have a pretty amazing experience with a big apex predator on a Caribbean coral reef because these guys do this, right? And I would argue that that's a very valuable experience, and one that divers are willing to pay a lot of money for. And so there's a value to Nassau grouper beyond a fisheries resource. There's a value to Nassau grouper and grouper in general, large apex predators on reefs, because they are what we think of in terms of a healthy coral reef. They like to take selfies. There's a selfie right there, right? They like to take selfies, and so there's a derived benefit from these things that's outside of just a fisheries resource and protein on a plate. All right, so if we take a step back, here we are in the Cayman Islands, 3 islands, Grand Cayman, which we are sitting at right now, and the 2 sister islands of Little Cayman and Cayman Brac. Historically, there were as many as 6 to 8 different spawning aggregations of Nassau grouper, 8 if you include the offshore banks of 12 Mile Bank. And all of these, by the 80s, had been fished to the point where things were really starting to hit the fan. And that's when the Department of Environment got involved. And Gina, who you just heard speak, not only administrator, she's also a scientist. She was involved in some of this work. A lot of the people that are still with the Department of Environment were involved in some of this early work in the 80s to create what is arguably the seminal study on age and growth for Nassau grouper. And this was based on the fishermen coming to the Department of Environment and saying, hey, help us. There's something going wrong with our fishery. And in addition to doing age and growth through otolith work, also looking at catch per unit effort and catch from the aggregations and demonstrating that, yes, in fact, catch per unit effort was declining. And remember, with hyperstability, if you can see catch per unit effort declining, it means things are really at the bottom end of where you want to be. Things are starting to decline. And by the mid-90s to early 2000s, all of these aggregations throughout the Cayman Islands were largely considered defunct. There was essentially no fishing left that was worth doing at those aggregation sites. But in 2001, an aggregation was effectively rediscovered on the west end of Little Cayman. And at that point in time, when discovered, that aggregation was roughly 8,000 fish. So the largest aggregation of anywhere in the Caribbean that we know of, and it remains to this day the largest aggregation for the species. And it's a unique resource, not just because there's a lot of fish there, but because it allows us to ask questions about what a healthy population of Nassau grouper should look like and how it functions. And that's critical if we're hoping to recover the population, not just in the Cayman Islands, but everywhere in the Caribbean basin. We have to get underneath the hood. We have to understand the mechanisms that drive population recovery in the species. So, in terms of the Cayman Islands, this was discovered in 2001. By 2002, there was an alternative year fishing law passed to reduce mortality, protections of those aggregation sites, and not just the West End, but every aggregation site, current and historic, throughout the Cayman Islands. In 2003, there was a fishing ban placed on those aggregation locations, and we did science associated with this work, and ultimately, as of this year, and Minister Panton mentioned this, grouper legislation was passed. And in brief, no take during the spawning season, bag limits throughout the rest of the year, charitable bag limits throughout the rest of the year, no spearing, no trapping, I would argue the most progressive set of grouper legislation in the Caribbean Basin. So that's good news, but how did we get here? How did we move over this last decade from just putting in place protections to putting in place some of the most progressive management actions for the species anywhere in the Caribbean Basin? And in order to get there, we built the Grouper Moon Project. And this is a collaboration between the Reef Environmental Education Foundation, which is a citizen science-based marine nonprofit, and the Cayman Islands Department of the Environment. And together, this collaboration set about to do the science necessary to understand the processes driving both recruitment, population recovery, productivity, starting to get at this mechanistic underpinnings of how aggregations function. So I'm going to be talking about the diversity of research projects that we've been doing as part of the Grouper Moon Project for the rest of the talk. And in order to do that, I think it's worth pointing out we're starting with the Nassau Grouper life cycle so that I can point to the different areas of the life cycle that we've done work on. So in terms of the life cycle, we have Nassau Grouper. If we start with recruitment, here we go. Young juvenile Nassau Grouper show up on recruitment habitat, nearshore habitat, macroalgae and turtle grass. And over a course of about 5 to 8 years, those young fish turn into big fish that get reproductively active. And by about 7 to 8 years, they're fully recruited to the population. And at that point, they aggregate to reproduce. And all individuals only reproduce during the winter spawning season, and they release their gametes. Each reproductive age individual has ballpark 1 million or more eggs. So over 1,000 fish, and you get billions of eggs released over a very short window of time during the spawning season. Those eggs get spewed forth into the deep briny blue, and they circulate around for about 30 to 50 days, where they turn into settlement-sized larvae, and then they start the process all over again. So that's the Nassau Grouper life cycle. So in order for us to do the work necessary to figure out whether or not and how to manage the species, we need to know a series of basic questions. At the very start of this, we had questions associated with how do aggregations work? Simple questions like, where are the fish that are showing up at these aggregations coming from, and where do they go home to afterwards? What proportion of the population are aggregating? These questions allow us to ultimately make decisions about how to collect information on the population to manage it more effectively. In order to do that, we need to know how aggregations function. Once we do that, and we come up with methods for surveying the population outside of the fishery, we need to figure out how many fish are in the population. Now remember, we can't rely on catch per unit effort or data from the fishery. And caimans for 2 reasons. One is because they're hyperstable, so it doesn't reflect the population. And 2, there is no fisheries data collection. It used to happen at the spawning aggregation sites, but there is no spawning aggregation fishery anymore. So we don't have fisheries data. So we need to collect information on the population outside of the fishery. And then finally, we need to get into the black box that is eggs released out into the deep briny blue to new juveniles recruiting to the reef. What happens in between that huge chunk of the natural history for the population, and what does that tell us about expectations for things like population recovery and what we might expect to happen through climate change moving forward? Because we're all going to have to deal with climate change, and it sure pays to know what we should be expecting in a population moving forward, given that it's coming, even to us. All right, so let's start with each one of those in turn. How do aggregations work? Some of you have probably heard me give this talk before, so I'm going to briefly go through this. There are spatial questions, right? So we want to know where the fish are coming from and where they're going home to afterwards. At the start of this project, at least some of the local community were making the case that these fish are coming from elsewhere, other islands, Haiti, Jamaica, all over the Caribbean Basin. And so we need to have an understanding for whether or not and to what extent that's true, or are these local Caymanian fish, and what proportion of all fish around the Cayman Islands are going to that aggregation site. And so we ring the islands with hydrophones. These hydrophones are little acoustic listening stations that can detect the pings of acoustic tags that we implant in the abdomen of fishes. These tags are uniquely identifiable. When a fish swims by a hydrophone, the hydrophone records that information, and we subsequently then go pick it up, download it to our computer, and we have a record of how the fish move around the islands. So we tag fish both at aggregation sites and at various sites all around the island when it's not during aggregation season. And this is just some slides of that. So each of these red dots here, this is Little Cayman, this is Cayman Brac, represents hydrophone locations. On Little Cayman, they're aggregating down here on the west end of the island. They used to aggregate up here on the east end. Over on Cayman Brac, they aggregate on the east end of the island. The hydrophones are on moorings that look like this, and these are just pictures of us tagging fish at the aggregation site off of the RV Seakeeper, which is the Cayman Islands Department of Environment research vessel. Okay, the way I'm doing this talk is I'm giving you the results, or the conclusions, and then I'm going to show you the evidence. So our results and conclusions from this work is, effectively, all fish that are reproductively active in the population go to the aggregation every year. They're all there. It's a complete population bottleneck during that season of time. Overfished populations aggregate longer. As you depress a population, as you remove more fish from the population, the few remaining fish that are in the population spend longer and longer and longer at the aggregation site. And why that matters in a management context is if you allow fishing at those aggregation sites, though you have fewer fish in the population, you're going to have longer access or time to get at those few remaining individuals and be able to catch them. And then finally, fish visit multiple aggregation sites. Some of the fish in this population travel 500, 600, 700 kilometers on an island that's 11 kilometers long. So they will move all over the place, visit multiple aggregations, but ultimately coalesce at a single one. And in a regional context, if you allow fishing on aggregations, when you harvest one particular aggregation to a point where it's very low, those last few remaining individuals are going to pick themselves up and go to another aggregation site. And that regional coalescence, again, gives you more and more access to the few remaining individuals in the population. All of this is the mechanistic explanation for why hyperstability happens in aggregating populations. These are the reason why we get hyperstability. And they are also the reason why, generally speaking, fishing aggregations is not a good idea for all of those reasons. Okay, how do we know this? Well, I'm gonna animate the data. So here's Little Cayman. Again, the solid circle is the aggregation site on the west end of the island. The dashed circle is the old historic aggregation site. The graph up top is full moon, goes to new moon, and the red line is real time moving forward. You're seeing those little blue and various colored dots moving around the island with red arrows. Those are fish moving around the island. So you're seeing real time sped up as we come up on the January full moon and beyond. And about 3 to 4 days after the full moon, every single fish in the system is there. They go and they visit the old historic aggregation site, right? This is regional coalescence. But come night of peak spawning, they're all there, all of them. So there's every single fish in the system goes to spawn, but they're visiting multiple aggregation sites. So even protecting one site, but not the aggregation network, you're going to do it wrong, at least a little bit. Okay. If we shift over to Cayman Brac, we have the exact same experimental setup, but now east end of the island is the solid circle. That's where fish aggregate. You'll notice that I started the animation. This is 2007, so it's a different year. Previously, it was 2006. I started the animation a little bit earlier. In 2007, they spawn in February after the full moon. But look at how much time they spend at the old historic aggregation site before they ultimately start to make their way to the new current aggregation site. Lots of movement. That maroon fish, that's the one that circled the island roughly 8 times during the spawning season this year. So we thought at the beginning of this, well, they're probably Caymanian fish, but they just make a beeline to the aggregation site and then go back home, and they do not. They move a lot. All right, but come night of peak spawning, every single fish in the system is at that aggregation site. So the interesting point here is that not only are these fish aggregating just like they did over in Little Cayman, despite the fact that the population is probably a fourth or 1/5 of the size of the one over on Little Cayman, but they're all doing the exact same thing. So it's not like aggregating behavior is breaking down in this population. And in fact, the converse is true. It's picking up. So if I shift over to the Little Cayman and I just look at the relationship between the amount of hours spent at the aggregation site within a spawning season on the y-axis against the fish size of those fish that are tagged, you can see that there's a relationship between the size of the fish and the amount of time the fish spends at the aggregation site. Bigger fish aggregate longer. Right? This is important because those bigger fish are the more fecund fish. They're the ones that have more eggs. If you're talking about an 80 centimeter fish versus a 50 centimeter fish, that 80 centimeter fish has roughly 10 to 100 times more eggs than the 50 centimeter fish. So the vast majority of the total egg production in the population is due to the relatively few old females in the population. Those are the ones that are producing the eggs. And not just a lot of eggs, but they're also producing better quality eggs. By and large, older, bigger females produce eggs with higher fat content. They're better able to survive starvation. Those eggs grow faster. So those are the ones that are responsible for your productivity and population. And that's why the Cayman Islands Department of Environment and the Cayman Islands put in place legislation for slot limits. You can't catch the babies before they reproduce, and you can't catch the big old females. Big old fat fecund females, the ones with all the eggs, right? Okay, so this plays out in the real world when we look at Little Cayman, but if we jump over to Cayman Brac, we see the similar relationship for size, but pound for pound, size for size, fish are spending longer at the aggregation site on Cayman Brac. If we take data from the U.S. Virgin Islands, this is from Rick Neiman's group, this is a population that only has 500 Nassau grouper. So we've gone for, at the time we did this study, there was roughly 3,500 fish over in Little Cayman, down to an aggregation of about 500 fish, as best we can guess. An 80 centimeter fish is spending 200 more hours at the aggregation site on a smaller aggregation. So as you decrease that aggregation size, those few remaining individuals are spending longer and longer hours, more and more hours at the aggregation site. And this gets to that notion of, well, if you allow fishing to take place, you're going to be doing pretty good at taking those few remaining individuals because you just have longer to get access to them. Okay. So we now know that and they're all Caymanian fish. That was the other big upshot, right? So none of these fish are coming from anywhere else. We had never had any fish do any abyssal migration, both through visual floy tagging or through acoustics. So we know that all fish go to the aggregation site. It's a complete bottleneck, population bottleneck for reproductive aged individuals. And we know that all those fish on the west end of Little Cayman are only Little Cayman fish. They're only Caymanian fish. All right, so that's great news because it allows us, if we can do an active and accurate census of the number of fish that show up at that aggregation site, we can completely census the reproductive age population. Right. So we can do that with tagging. I'm also going to talk about two different other approaches that we use to census a population. One is through a citizen science marine nonprofit outside of the spawning season. So that allows us to get at estimates of density and abundance throughout the rest of the year. And also some passive acoustic work that we are doing, and it connects really nicely with lots of other research programs throughout the Caribbean basin. So not only can we get indices of abundance from it, but we can also relate our indices of abundance to other programs throughout the country and gain inference in a collaborative way. Okay, so the tagging. So you're seeing a video of a tagged Nassau grouper with a floy tag in it. Conclusions before evidence. So we have demonstrated over the last decade that aggregations can recover, and can recover quite quickly if they're given adequate protection. Little Cayman, as a population, has more than doubled over the last 9 to 10 years. Brac, based on fairly limited data that we've got, appears to be at least stable in terms of the population. On the other hand, Grand Cayman has continued, if anything, to decline, and not just by a little bit. Grand Cayman population of Nassau grouper continues to drop. All right, so what's the data, what's the evidence that we've got? So we do floy tagging at the aggregation site on Little Cayman. We tag a very small proportion of the population. Then we send divers into the water, and we do counts of tagged, untagged individuals over and over and over again. And those counts allow us to get an estimate of the proportion of the population that's tagged. If we know that proportion, and we know how many tags we put into the population, we can estimate the total number of fish that are there. Right, so it's a simple marker capture method. We can do that year over year. We've done that since 2008. We incorporate that into a fancy Bayesian modeling framework to estimate population growth through time. And so you're looking at the estimate of abundance where you've got the median estimate in black, and then the gray bars there represent our confidence or our uncertainty in those estimates. Squint, draw a line through it, and it's fair to say that if anything, there's an exponential increase in that population through time, moving from, in 2008, roughly 1,800 individuals, to 2015 and 16 when we're closing in on 5,000. So a big growth in the population. And down here on the lower right, you're looking at a histogram that shows the difference in growth through time. So it's a growth rate. So there's quite a bit of variability, but on average, the population is growing about 10% a year. And that variability, I would argue, is driven largely by variability in recruitment, and we'll get to that a little bit later on. But it's important, that variability is important because it allows us to set expectations for population recovery moving forward. Okay, that's the tagging work. We can also lean heavily on the Reef Environmental Education Foundation's Reef Fish Monitoring Program. This is a program that's been in place for the last 30 years. Fortunately, in the Cayman Islands, there's lots of diving and lots of people writing down the different types of fish that they see, as well as reporting relative abundance. This data it happens year-round, and while it's categorical in nature, it's also a large enough database. There's enough data to actually give us fairly highly resolved estimates of abundance through time. And so what you're seeing is, since 1995, the estimate of abundance of Nassau grouper on home reefs from Little Cayman in yellow and Grand Cayman in blue. And this red out area here is since protections have gone in place. And so what you're seeing is that the reef data largely agrees with the tagging data and that the population is growing through time. And in fact, this is on the log scale. So a linear increase on the log scale is an exponential increase, right? So you're seeing an exponential increase in the abundance score based on reef data for Little Cayman. And conversely, for Grand Cayman, you're seeing the opposite. So I would argue that Grand Cayman presents a real opportunity to address the management problems that are here to start to recover the population. We know from Little Cayman that if you protect them, and you protect them appropriately, you can get rapid response in the population. We just need to figure out how to do it here. We need to figure out how enforcement can happen in order to make this population turn the corner. All right. And then finally, we'll talk briefly about acoustics. We put a can in the water that's got a hydrophone and a heck of a lot of batteries, and that thing sits there year-round and records 20 seconds every 5 minutes acoustics. It turns out fish are like birds, or at least grouper are. They call, they vocalize, especially when they're interested in sex. They'll do a lot of calling, right? And so the fact that they call allows us to develop an index of abundance by recording how many times you hear calling at a site. And this is the same technology that that's being used in the United States. It's being used in the Virgin Islands and Puerto Rico. Lots of different people are using this exact same technologies. And it's cool because not only can we collect information that gives us an index of abundance, but we can compare it in a regional context to what's going on with the species. And that allows us to make some really interesting inference about what's driving the population dynamics for the species. So we have these at each of the aggregation locations throughout the Cayman Islands. We also have one at a non-aggregating site up here. That's our control. And so from this, we can generate these graphs. Here's a for instance. So this is data from the west end of Little Cayman. The y-axis is calls per day, and this is across dates. The spawning season for this year was in the end of February, so you're seeing real peaks in calling, and we can translate this ultimately into some version of an index of abundance. Now, how we do that specifically, we're still working on the details. And it involves things like having a baseline. So this is the non-aggregating site. And you can see that even at the non-aggregating site in the end of January and February, there's a peak in calling. So they're not just calling at the aggregation site, they're calling while they're moving to the aggregation site. So, you know, they're cruising for the ladies, and they're booming as they go. So we need to understand that baseline, because if you were to just look at this, you'd go, hey, this is an aggregation site. I'm hearing Nassau grouper calling. No, it's not. It's just they're calling while they're moving. So we need that baseline. Okay, so that's assessing the population for looking at change through time. We also need to know something about the natural history of the black box, that is recruitment, starting from eggs and leading all the way to those newly baby Nassau grouper settling out into recruitment habitat, which is the nearshore habitat. And we can have addressed this in a multitude of different ways, but really what we're asking is, and this is cooler if it's nice and dark and you can see the video, but if you can see from the background, it looks like a giant strafe that's coming up, and you've got roughly 30 to 40 fish all releasing out into the deep briny blue huge amounts of gametes and sperm. This site will start at 120 feet of visibility, and right at the point it gets pitch dark, that visibility can go down to 5 feet or less, and it's all sperm. You are literally swimming in a sea of sperm. And so the reproductive potential there is massive, and it's because all spawn happens at that point. All of their eggs are being released over a series of about 3 days. We can track those eggs, and it gives us a really unique window into what happens in the early life stages of the species, which is the gauntlet, right? It's where a lot, if not all of the mortality, not all of it, but a vast majority of the mortality happens. So we can start to ask questions about what's driving that mortality. And in a management context, is there something we can manage, or can we at least set expectations for how a population recovers based on our understanding of the mechanisms of early life history, growth and mortality and survival? All right, so we do this in a couple of different ways. We've been monitoring recruitment around the Cayman Islands using light traps and snorkel surveys. We did light traps for about 3 years. We've been snorkel surveying every year since 2006-ish. We've been setting out drifters. These are drogues that you put in the water, and they follow the water column that the eggs are spawned into. You also have on top a ball that floats at the surface, a GPS, and a satellite phone, and it reports the location. So we can track that body of water where the eggs go into. And we're also, because we have access to those eggs, we're also looking at the early life history and demography of the species as it grows in that first window of time, that first major mortality window. Again, conclusions before evidence. Recruitment is highly episodic, and highly really should be emphasized here, right? Highly is in most years we find nothing, and then 1 year we find a heck of a lot. So hugely variable recruitment. This is important because if we don't know about this, we might not be able to set expectations for recovery. And so you could imagine wherever you are in the Caribbean, you put in place reserves and the population doesn't respond, and somebody goes, well, look, your reserves aren't working. Well, that may not be true. It's just that you just haven't got new fish showing up in the system yet. Fish spawn at times in locations that lead the larvae that are produced ultimately back home. And this is important because what it means is your local adult reproductive aged individuals are the engine for population productivity and ultimately recovery. Your adults are important to you, and if you don't have them, you won't get new babies showing up. And then finally, a warming ocean may be ultimately really, really bad news, at least for Nassau grouper, and at least here in the Caymans. So we'll talk about each of these. Okay, for monitoring recruitment, again, snorkel surveys and light traps, did this for years. And you are looking at a summary and all of the complete amount of data that we've got right here, this guy right here. That is the only Nassau grouper we caught in our light traps. We set them out on Little Cayman, we set them out on Brack, we set them out on Grand, and we caught one over 3 years. So not a lot of recruitment, right? Through snorkel surveys and reports from local dive masters, we would get maybe one juvenile sighting reported every 3 years or so, and literally one fish. So we're dealing with a very data-limited situation here, right? With the exception of 2012. 2012, we started getting reports from local dive masters. Hey, I'm seeing Nassau grouper juveniles all over our moorings for our boats, and we got calls from local fishermen saying, hey, I'm in the back bays, I'm seeing schools of Nassau grouper, and you're on the phone going, schools? They don't even do that. How do you mean schools? Well, it turns out they do. We had a huge pulse of recruitment of Nassau grouper, and not just in Little Cayman. We had it in Brack, and we had it in Grand, and if you looked in the broader Caribbean basin, there was a big pulse in Bahamas, and even places like Nevis started having Nassau grouper show up where they had not been seen before reliably. So I don't know what the process is, and perhaps at least a little bit the process is not all as important as to say that we know that pulse recruitment in the species is a really big deal, especially for the Caymans. And understanding that and managing for that is important. All right, and probably why that is because, so this is a map of connectivity. Now you've seen this, Bob Cowan's work, it's been done about 1 billion times, right? So this is one of the more recent publications looking at Caribbean connectivity. This wasn't for grouper, but it doesn't really matter. Connectivity is connectivity. So here's us in the Caribbean basin, and let me get my cursor up, and here's us in the Cayman Islands. So the heat of the line indicates the amount of connectivity between these regions, and you'll see that in the Cayman Islands, really the only connectivity we've got, and it's pretty darn low, is between here and Cuba. And that's it. Right? The Caymans are really isolated. Again, that's more reason for this notion that your local adults are important to you, because you're not getting a lot of external support, and where you are getting external support is from Cuba. And we've all seen what Cuba, Nassau grouper, they do a really good job of documenting, the last 5 decades worth of for Nassau grouper. So you're not getting a lot of external support from Cuba, unless things have changed. So this is probably why we're getting that pulse recruitment, is every once in a while, all of those eggs that are released over 2 to 3 days somehow manage to make it back, which is remarkable that they do. Right? The ocean's a big place. So how do they make it back? Well, sorry, we should say, we study this in a regional context by using drifters. Again, we talked about what drifters are, so we can release those at the spawning site. And this is important because those maps that I showed you, and all the maps that are generated like that, are based on drifters that float around and are released, usually from plains in the middle of the Caribbean. And none of those are released at the point at which eggs are released right coastally on shore. So we assume that those drifters floating around approximate connectivity among islands, but we don't know, because none of those drifters have been released at a shelf edge where fish are releasing eggs. We can do that. We can do that here because we know exactly where all reproduction happens for the species. So we do that on Little Cayman, and this was, first did it back in 2008. We've done that every year since. And, oh no, media not found. All right, that's okay. Take my word for it. What you would see if this moving were working is you would see nights leading up to peak spawning, you're going to see these drifters basically make straight line movements away from the island. But on nights of peak spawning, those drifters do something fundamentally different. They gyre. They get caught in eddies. And this has been consistent across all the years that we've done this now. More often than not, on nights of peak spawning, when you release those drifters, they get caught in these little eddies that keep them close to home. Now, we don't know what the mechanism is, how in an evolutionary context they have picked these nights. We don't know what that mechanism is, what the cue is, but it's nonetheless very clear at this point that those fish are choosing to release eggs on nights when those eggs stay close to home. Okay, so this is just data from this last year, hot off the presses, back in February of this year, actually. So we released drifters on 2 nights. They spawned on 2 different nights. The first night, we released a drifter, and 3 days later, that drifter is roughly, 15 miles up a Rum Point from Little Cayman, right? And then the second drifter on the second night, we released it after 3 days. It was not more than about 20 miles from Little Cayman. It basically just did loops on the aggregation site for 3 days. So, again, getting caught in those eddies, so sticking to that pattern. And I think this connection between Little Cayman and Cayman Brac is really important because it's clear evidence that Little Cayman, that big population, is the one that's going to be providing recruitment to this island. Those grouper that are releasing eggs are sending their larvae over to Grand. And this is why Little Cayman matters even if you're a fisherman on Grand or on Brac. You need that population, because that population is the one that's going to give you eggs and new baby grouper showing up on the reefs so you can rebuild the population here. Okay, prove it. So you can imagine people going, and probably some of you in the room are going, yeah, but those are drifters, they're big socks that hang in the water. They're not larvae. They're not eggs. How do you know that those things even remotely track what's actually going on with biology? And we heard this a lot. And so this last year we said, okay, well, we're at Scripps. We've got people that develop instrumentation all the time. We got a guy there, Jules Jaffney. He's just made these really cool underwater microscopes. What if we slap those things on the back of a net and tow it through the water? We go and talk to Jules. He's like, great, let's do that. Get up on the whiteboard, draw things out, send it to the engineers, and 2 weeks later, he's built the system. 2 microscopes, computers on board, flash every 1/8 of a second, so you get every 16th of a second, you've got imaging of anything that's flowing through that net. The computers on board are real-time processing those images and identifying ROIs, regions of interest, and saving those files to disk. This thing's crazy, right? Amazing. But you build this thing, and at least some of you have had some engineering experience. The first time you build something like this, if it works, you've won the lottery, right? You won the lottery. So we bring this thing down to Caymans, we throw it off the end of the Seakeeper, and it's hundreds of pounds, so it's not, I'm making it sound easy, and it's not all that easy. And then you find an intern, and you throw them off the back of a moving boat, right? And you say, hey, go look and see, oh, there's sharks by the way. Go look and see whether or not this thing is working, if it's flying right. So she does her duty, thank goodness. This is Tammy, and she jumps in the water, and she kicks out all the while worrying about sharks, and she catches video of this thing as it's flying behind the boat. We're doing roughly 3 knots, and lo and behold, this thing works. Not only is it reporting data to us, but it's actually flying the right way, the net's working, everything about it is doing the right thing. This video is going really slow. Oh, there it is. Out of the depths. You know, at first it could be a shark, you don't know, but it's not, which is good. All right, so there you go. So this thing's flying, it's catching images. So totally works, and this is awesome. And captures roughly 3,500 images per 30 minutes towed, so it's saving things just rapidly on the fly. Over a 4-hour window, we've got, I don't know, 400,000 images of things in the water. Really high resolution images in color of things that haven't been impacted by nets. So pretty amazing stuff. So here's some of the stuff we caught. Here is a ketognath. You guys know what these are. Some of you probably do. I am not a plankton biologist, so I think this is all cool, and I don't know what most of it is. But so you get this big, long creature, and these things are like the velociraptors of the sea, right? When I'm doing this, but it's this, right? But these things eat everything that comes in their path, and so here's a ketognath who has apparently very recently eaten some poor copepod that's now down its gullet. And I point out ketognaths because here's a ketognath with an egg right in front of it. At first we thought maybe it was in its stomach, but I don't think it is. But in any case, you get an idea of the size of the ketognath relative to the eggs, and this is a predator. So predator-prey interactions in the plankton are equally important. You know, if you think about them as like dinosaurs as opposed to tiny things, these predator-prey interactions are important in that early life history. All right, who knows what that is? It's pretty. Anyone? Looks like jewels, right? Okay, how about now? Egg sac of a copepod. Right? So we found that egg sac of the copepod person, like, what the heck is that? Looks like a thing of grapes. Natural history is amazing. All right, we get diatoms. We get this thing. I don't know what it is. Anyone know what that is? The hairy thing. We get eggs. So this is just really amazing. I've compiled this image. So this is not one image. This is a bunch of things stuck together. But what I think is really cool about this is you can actually get developmental stage. The resolution is such that you can see the development. You can actually literally count the cells here. You can see development in action as we're toying around, all the way to larval fish. So really cool stuff. And so we do this. So what you're seeing is night of peak spawning, we release 3 drifters. Those are the green things. The black dot represents the mean of those 3 drifters moving forward in time. You throw the net camera over the boat, and then you're looking at the path of the boat. The size of the red circle is the number of egg-like objects per unit time. This is after spawning, right? So we throw the drifters into the spawn, and we start to track those drifters with the boat, and you see this real time what the plumes of eggs look like. Some areas higher, some areas lower, and this is just like the first cut of this stuff. We can start to really hone in on processes of development, mortality, survival, and relationship between eggs and ultimately grouper larvae and the things that they eat and the things that eat them. At the 30,000 foot level, we can say, yes, drifters are tracking eggs. And that's an interesting thing, but it's not remotely the most interesting thing about this data. So we can really start to ask questions about what is influencing early life history, mortality, and survival for the species. And I think that's really cool, and it's going to allow us to make some really neat, nice insights into the ecology and the ecosystem that Nassau grouper exists within. Okay. How do you know you're looking at grouper eggs? We see eggs, but how do we know they're grouper eggs? Well, we can actually collect these eggs and we do genetics on them. So this is a histogram of the size of eggs and the frequency of eggs. And you'll see that there's multiple peaks, suggesting that there's multiple species that are spawning. And genetics says, yes, there are. We got eggs from Creole wrasse, red band parrotfish, yellowhead wrasse, barjack, not surprisingly. But here's Nassau grouper way out here at the end. Where's my cursor? So this is our peak of Nassau grouper. So this is really cool news because not only are we collecting information on Nassau grouper, but lots of other species as well. But we can clearly identify what constitutes Nassau grouper eggs from these imagery based on genetic work. There's going to be more about this later on, and I'll briefly introduce some other folks that have been talking about various aspects of the work I'm presenting today. And then finally, because again, we're at the spawning site, we can go out with bags and we can actually collect eggs from spawn that happens. So we can see a spawn, we go out, we take our Ziploc bag and we capture those eggs, and we can bring them back to the lab. We work with CCMI, the Central Caribbean Marine Institute, to raise those eggs in the lab. And we can ask questions about fertilization rates, growth, and survival. of those eggs. We catch them at the aggregation site, spawn in situ in the wild, bring them in and see what that early life history process is like, and also how it's influenced by things like temperature. And it turns out that temperature is really influential. Just a few degrees difference in terms of heating can have dramatic impact on time to mortality. As you increase from 24 degrees to 26 degrees centigrade, you roughly cut in time by half the amount of time larvae can survive. That's important because when Nassau grouper spawn, if we look at the temperature line here, this is the average sea surface temperature in the Caymans, January and February, that's the trough, right? That's when it gets coldest. So if we know already that warming temperature from 24 to 26 dramatically increases the rate at which these things die, and we're already sitting at roughly 25 when they spawn, any increase in sea surface temperature is going to cause pretty dramatic impacts on how these animals function and ultimately die in the early life history. And we also know that within the next 100 years, we're going to get sea surface level changes of anywhere between 2 and 4 degrees C. Undoubtedly, that's going to have a dramatic influence on how this species performs at recruiting. And it doesn't matter if you're here, it's probably everywhere in the Caribbean basin. And we need to know this in a management context. Why we do this research, it sounds all esoteric and sciencey, but we do this research because now we can set expectations for what happens into the future of not just 10 and 15 years, but 100 years from now. Where are we thinking this fishery is going to go? And this matters for things like food security, for the process of generating food for local consumption, for the local environment. And it should be pointed out here that 2012 came from fish that were spawned in 2011. Who here remembers 2011 if you were living in the Caymans? So that was the coldest year it was one of two of the coldest years on record. The only other one that was colder was 73 in terms of winters. That year, coincidentally or not, I don't know at this point, but that year was the coldest year they've had for a long, long time. Okay, so here we are, spawning and studying spawning aggregations in the Cayman Islands is allowing us to access all axes of this life cycle for Nassau grouper and start to gain insights into what's going on. And from this, we can do really cool things like build fully size and age structured assessment models. We can do that, at least for Little Cayman, and hopefully from some of the other islands as well. This also allows us to ask questions about, for those areas that we don't have all this data, how well do data-limited assessment models work? So we can move just beyond Cayman Islands and say, in general, how well do we manage this population and what models can we use in order to do things more appropriately, given this hyperstability problem. We can also focus on coordinating our management. More and more in the Caribbean, governments are working together, especially for particular species or resources that are important to us. And this information feeds into the process by which we do coordinated management for aggregating species in general. Ultimately, all of this is aimed at restoring healthy fisheries. We're not just about conserving a fishery. We're about making it so that there's enough fish for divers to see, enough fish for them to fill the ecological role that they're supposed to fill, and enough fish that you can put fish on a plate reliably. That's where we need to get. And this research is aimed at doing that. None of this would have been possible without the dedicated staff of the Cayman Islands Department of Environment. They've been doing this long before we started the Grouper Moon Project. They're probably going to do it long after we finish the Grouper Moon Project. This is just some of them. That's not Phil giving the finger, by the way. He's showing you that he embedded a circle hook in his index finger. And if any of you have had a circle hook, look at it next time and think about what would be involved in embedding something into your finger. It's truly amazing that he managed to do it. And this was as he was swinging his bait around talking about how he's going to show everybody how to fish. So these guys make this work. And they're not just facilitators, they are part of the scientific process. They motivate the science that we do. So it's really a unique group of people that are dedicated to what they do and uniquely capable of doing those things well. So I really appreciate all that they've done. Very briefly, I've done highlights of a lot of different stuff. There are deeper talks and posters to be had on all of these different aspects. If you're interested in the marker capture analysis, Lynn Waterhouse is going to be talking about that on Friday at 11:45. If you're interested in the citizen science reef data, Brice is going to be talking about that today at 11:30. Drifter surveys and connectivity, Croy McCoy is going to be talking about that at 2:15 on Tuesday. The NetCam stuff, the plankton tow stuff, Brian Stock's going to be talking about that at Friday at 10:45, and then Lauren Arnold, who's an undergrad here with the lab, she's giving a poster about that and the genetics. Early life history and growth and survival, Allison Condomo, who is with CCMI, she's giving a poster about that. The regional coordination and management, Dr. Steve Giddings is talking about that at 10:30. And then the stock assessment aspects of this and the potential for stock assessment is poster bid given by Brian Cohen. So take a picture of that, you can revisit that. And with that, I'll just say thanks to the various funding agencies from LenFest and NSF, Disney, Guy Harvey Ocean Foundation, NOAA, all contributed to this project through time. And in addition to that, we've got various academic partners and not to be diminished, local partners, local companies and businesses like the Southern Cross Club and the Little Cayman Beach Resort and the nonprofit marine station CCMI. Lots of people have made this project function through time, and I appreciate all their support. And I'll say thank you.
Thanks for watching. We look forward to seeing you at the 70th Annual GCFI meeting in Mérida, Mexico on November 6 to 10, 2017.